Hey guys, well, we're on to a new chapter, and we actually kind of, over another hump, we're going to a new unit. And this is one of the most interesting units because you guys will be able to see things that are happening on you, things that you can do, because we're getting into genetics. So, just some things to start out, to get your mind going, thinking where we're going to be going. I got a few things. We know genetics obviously has the word gene in it. And genes control everything. They control what you look like. They control how you act. They control behavior, personality, intelligence. All these things are based on genes. Is it solely genes? No. A lot of things that we have to keep simple, we rely heavily on genes, but realizing that the environment has a lot to do with what you look like and how you act. So let's see what you guys can do. So I have a little sheet right here. It says, what genes do you have? And now that you're at home, you can actually go around once you figure out what you can have. Look at your brothers, sisters, parents, whoever is there that might be blood related to you to see what they can do and maybe they passed it down to you. So first question, can you roll your tongue? I can. See if you can do it. Some people can't. People that can do it struggle with the fact that some students will sit there and can't do it. They can't do it. It's in a gene. Other things. Can you bend your fingers like this? I can really do it well. Looks like I have like arthritis or something. So try that out. Can you do that? Look at your toes. I have shoes on so I can't do that. But look at that second toe. Does it extend beyond your big toe? If so, that's actually caused by a gene. What about the people? Can you do this? Some people struggle. They can't do the Spock, what it's called. Star Trek. How about fold your hands. Fold your hands like this and see which finger actually lays on top. Some people will actually put their left over right like I do, but then others will lay right over left, which feels awkward for me, but for some it is more common. Okay? So those are a few different things that I want you to see what you can do and then just test your other family members and see what they can do and see if you can do something that they can't. Since you came back, hopefully maybe you'd press pause. There are 7 words that are truly important for understanding what I'm going to be going through today and throughout this chapter. They're going to be in your vocabulary, but the sooner they get in there, the easier this will become. So what you're going to do, I want you to press pause, look at these words, and I want you to find a definition. Look for a definition on the internet. If you have your textbook, find it in your textbook. After that, in your textbook or somewhere, see if you can see it in context. Some classes, one class already did this today, but for the other 3, I would like you to see how they use these words when they assume that you know what the definition is. So here are just 7 words. Take your time, press pause, come back, I will be here. I will continue on as if it didn't actually press pause, but look these words up. They are truly important for understanding today's lesson and for the rest of the chapter. Go. Alright, welcome back. So the next thing, I'm going to relate meiosis, which is the chapter we just finished, to the new chapter, which is called Mendelian genetics, all around this great scientist by the name of Gregor Mendel. He worked with pea plants. Not that interesting, but this is how he got his start, as a botanist, studying the different characteristics of pea plants. He found that pea plants actually came in different varieties of things. They were genes. Genes were passed down from one pea plant to another pea plant. And he looked at 7 different genes. He would look at the flower color. He noticed they came in 2 variations. He would look at the pea pod itself and see it came in 2 different colors. He looked at the length of the stems. Some were long, some were short. Everything came in varieties of 2. Because what will happen is genes have 2 what are called alleles. Alleles is that variation within a gene. So the alleles that he saw were like purple flowers versus white flowers. He didn't know the idea of dominant recessive, which are then these 2 words. He did realize eventually that some alleles were dominant to others. Dominant does not mean more often found in a population, as contrary to what many people believe. It has nothing to do with the number of things in a population. 5 fingers is actually recessive. I bet you didn't know that. 6 fingers is dominant. So that already nullifies the whole idea that dominant means more in a population. Not true at all. So let's get into how to relate meiosis to this new topic. So if we look at meiosis just in these cells, if I start with a cell that has, I'm going to look at one chromosome. So if I say that my first chromosome here has on it a gene that has the dominant A, its homologous match, which I will change its color to, same size, same shape, different color, may have the recessive form of the letter A. This is what makes it homologous. They have different alleles potentially. Sometimes they could have the same, but in this case I'm going to make them different for the fact of this lesson. Then what I'm going to do is I'm going to go through the process of meiosis. So I'm going to replicate this, and when you replicate it, you make a sister chromatid. So which allele must be on this red sister chromatid? Correct, the dominant version, because you made a replicate of it. On the other homologous match, when I do this, it also has to have the recessive A. So when you go through meiosis 1, you separate into two cells. You now become a haploid cell that's a replicated state. So therefore, what would eventually happen, I'm not going to go through the whole phase, we just finished that chapter. You should know how to do this. In this cell, we could potentially have the blue X. So that means the recessive and the recessive. Over here in the other cell, because it's haploid, it has the other homologous match to it, still in replicated state. So I'm going to follow the alleles through the meiosis, which we haven't done yet. And finally, what would happen in meiosis 2 is this replicated chromosome would split into two non-replicated chromosomes. So you would have one non-replicated in each of these cells and one blue non-replicated chromosome over here. Recessive A, dominant A, dominant A. Okay, and that's where you'd end up. These would be sperm cells. Two of the sperm would have the dominant A allele in them. Two of them would have the recessive A allele in them. Important to keep this in mind when we go through what is called a Punnett square, which some of you from 7th grade may remember. Let's go through now what would an oogenesis kind of look like. So this person had a genotype, the male, since I just said these are sperm. They had a genotype, which was the word I asked on the previous thing you should be aware of now, had a genotype of big A, little a. Alleles have the different phenotypes. So without me telling you, I can give you a key. I will say, in this case, big A means you have unattached earlobes. Another thing caused by genes. They like dangle. Some people have it completely adhered to the side of their face. Others have it dangling. The recessive form, we'll say, is attached earlobes. So now you can check your earlobes and see what you would have. So this person going through spermatogenesis has this genotype. Big A, little a. They are heterozygous. For a phenotype, they are going to have the unattached earlobes because they've inherited the dominant form. It masks that recessive like it's not even there. They will exhibit the unattached earlobes. So they'll have, both earlobes will be unattached. So now if we go into the egg, and let's pretend that they also have a red chromosome, and they have a blue chromosome that I'm going to show immediately in the replicated state. I'll reverse it. I will say that on the blue one over here, it has the dominant A, so that makes that sister chromatid right there also have the dominant A. On the red chromosome, or the other homologous match, I'll say they have a recessive. So still, the genotype of the woman in this case also is dominant A, recessive A. Phenotype, still, she would have unattached earlobes. When this oogonium, or the starting cell of oogenesis, goes through, here's the end of meiosis 1. So what you would have is over here, there's the one homologous match. Over in the other cell, there's the replicated form of the dominant A. You can then foresee what's going to happen down here. These two sister chromatids will pull apart, and these two will pull apart. So since I'm on blue, I'll stay over here. Therefore, here are the egg cells. I'm not going to assume which one I'm going to make the polar bodies and which one I'm going to make the ovum. I'll assume that any one of these could be a potential egg. So here are potential eggs. Two of them, once again, have the dominant A. Two of them have the recessive A. So now, what we do is a Punnett square, which says I'm going to take any of these egg possibilities and say what is the probability that they could meet with any of the other sperm possibilities on the previous slide. So there are the sperms. There are the eggs. We set this up via a Punnett square, which is just kind of a matrix, if you use that in math, a matrix of the probabilities of how often they would meet together. So here's what a Punnett square could look like. I've lined it up so that I put the 4 possibilities of eggs along the top. I put the 4 possibilities of sperm along the bottom. I'm not going to draw the chromosome because I'm really only concerned with the letters on the alleles that were on the chromosomes within those eggs and sperm. So going back to the sperm, I saw that I had big A, little a, little a. So I'll do that. Over here, I'm not going to change letters anymore.